Hello, everyone, and welcome to this, our final webinar in our 2024 science series entitled Bright Breakthroughs, Real Stories of Beating Rare Disease. I'm Erica Berg, Director and Senior Editor for Custom Publishing at Science, and I'll be the moderator for this discussion. This is our fourth year exploring the challenges and successes in rare disease. Last year, we tackled advocacy. You can read more about those conversations in the Resources tab. This year, we've shifted our focus to what we can learn from personal stories of triumph. This year, we've covered mental health, caregiving, healthcare access, and workplace wins. You can check out those webinars on science.org. Now, we're turning our attention to the ultimate win, finding cures for rare diseases. While a lot of attention has been focused on emerging cell and gene therapies, which we'll definitely be talking about today, there are also other promising roads to a cure, such as repurposed drugs. But many obstacles still exist. The science continues to evolve, and there is also the issue of healthcare access. However, there are a few shining examples of how recent advancements are bringing actual cures to people living with rare disease. Before we get started, I wanted to give a few instructions about the viewing console. Just to the right of the video window is the Presenters tab, where you can find photographs and biographies of today's speakers. You can log into your social accounts during the webinar to post updates. On X, you can add the hashtag #ScienceWebinar. Now, we pre-recorded this webinar a few weeks before this broadcast, so we won't be able to take live questions from viewers, but we'll do our best to cover the topic as comprehensively as possible in the limited time we have. You can feel free to leave comments for us in the Ask a Question tab to the right, and you can reach us if you're having any technical issues. You can also email feedback to webinar@AAAS.org. Finally, a thank you to Foundation Ipsen for sponsoring today's event and this series. I'd now like to take the opportunity to welcome our fantastic panel today. I'll give each of them a chance to say hello and introduce themselves. First, I'd like to welcome Michelle Werner. Hello, Michelle.
Hello, how are you? Thank you for including me today.
Well, tell us a little about yourself.
So a little bit about myself. So I'm the CEO of a flagship pioneering company that's called Alltrna. We were founded in 2018. We're still preclinical right now, but we're really focused on unlocking the biology of tRNA or transfer RNA to treat a number of different rare genetic diseases. And one of the things that's super cool about our technology is that we can use a single one of our medicines to treat hundreds, if not thousands, of different diseases. And when you have rare and ultra rare diseases, you can, clearly appreciate the potential impact that that might have to bring some new innovation to lots of patients who may not otherwise have that. So that's what I do in my day job. I'd say the other part about myself and my connection to this topic today is that I'm a wife and a mom, and I have 3 kids. One of my children is 14 years old. His name is Caffrey. And about 4.5 years ago, on his 10th birthday, he was diagnosed with Duchenne muscular dystrophy, which is a rare genetic muscle wasting disease that's progressive in nature. And today there is no cure. And so I've been sort of thrust into the rare disease community myself and really passionate about bringing novel innovations to patients and families that are pretty similar to my own. So it's a pleasure to be here and happy to talk about our story, both personally and professionally.
Thank you so much, Michelle. Also with us, we have Bill Hobbs. Hello, Bill.
Hello, thank you for having me.
You want to share a little about who you are and what you do and why you're here?
Sure. Yeah, I'm lead the clinical development programs for hematology at a company called Vertex Pharmaceuticals. And the area in particular that I've worked on for a number of years is in sickle cell disease and beta thalassemia. And we've recently developed a potential treatment called exa-cel, which is a CRISPR-Cas9 gene editing program for the treatment of transfusion-dependent thalassemia patients and severe sickle cell disease with recurrent vaso-occlusive crises, which is now known as Casgevy. I've been involved in hematology and in sickle cell disease for well over 20 years now. I originally started from a research standpoint, being interested in being able to leverage technology to develop genetic therapies for diseases. And for a long time, sickle cell disease and beta thalassemia had been model diseases that everyone thought that at some point we would be able to develop a genetic therapy for. Took a very long time to get there. I think what changed for me as I was developing my career was moving from a scientific focus into one that was a more patient focused. And for me, that happened with exposure to people living with sickle cell disease and beta thalassemia in medical school. And that changed everything. And I think that's a common theme for people that get involved in rare disease. It's really the patients and families who are affected that drive all of us. And what becomes very clear in that setting is that it's very difficult for people to get access to appropriate care and equitable access to care. The research resources are sometimes limited, and the treatment options are as well. And so the motivation for me along the way has been to merge that research interest to develop tools that can be treatment options for patients who really have very few, if any, other options. And that was certainly the case in sickle cell disease and beta thalassemia. So this has been, I think, for us at Vertex, a very rewarding journey in terms of developing what could be a treatment option for patients who have a severe and life-limiting disease with very few other options.
Yeah, wow. Thank you so much for being here, Bill. And finally, I'd like to welcome David Fajgenbaum. Welcome, David.
Thanks, Erica. I'm so happy to be with you today. My name is David Fajgenbaum. As you mentioned, I run a center here at Penn called the Center for Cytokine Storm Treatment and Laboratory, where we do in-depth immune profiling of rare inflammatory diseases. I first got exposure to rare inflammatory diseases when I was a medical student here at Penn, and I became very ill with a rare inflammatory disease called Castleman disease, and nearly died 5 times from this horrible condition before eventually discovering a drug called sirolimus that had been around for decades for other diseases, but had never been used before for Castleman's. I tested it on myself, and it's now been over 10 years that I've been in remission. And during this 10-year remission, I've just been completely on fire trying to find as many more uses for existing medicines as possible. Here in our lab, we've identified over a dozen additional drugs for diseases that they were not initially intended for. And then 2 years ago, launched a nonprofit called Every Cure to do this at scale, to leverage the power of artificial intelligence to look across the world's biomedical knowledge, every drug, disease, gene, target, protein you can imagine, and quantify the likelihood of all 4,000 drugs to be able to treat all 18,000 human diseases, and then take the most promising opportunities forward. And I'm super excited to be with you guys today and to share more about this work to try to save lives with existing medicines.
You're an inspiring bunch, I must say. So let's dive into our discussion. We're going to start a little bit talking about the science. Bill, I was going to start with you. Can you describe in a little more detail sort of the journey behind the development of, I believe it, I'm probably going to mispronounce it, but is it Casgevy?
Yeah, thanks. It's Casgevy.
Casgevy. Okay.
The name in development, we call it Exocell, which is actually short for exogamglogene autotemcel, which is even a more mouthful. But I've mastered that over the last couple of years, so I can rolls off my tongue easily. I think for a lot of rare diseases, and sickle cell and beta thalassemia are like this, which is that we've known about the diseases and the underlying cause for a long time. Sickle cell disease was first described over 100 years ago clinically. And the causal biology of it being a mutation in the beta globin chain of hemoglobin has been known since the 1950s. We've also known since the 1940s that fetal hemoglobin reduces the impact of sickle cell disease in research that was published by Janet Watson in 1947. So we've known about these things for a long time. What I think changes over the years is as we understand the biology, we get more and more information about understanding what potential targets we could actually attack for potential treatments and what tools we could use for those. And so what I think really changed for us at Vertex was about 10 years ago, the first description of CRISPR-Cas9 gene editing, which could be potentially used as a therapeutic. And that means that, you could potentially identify a precise and specific place in DNA and make a very precise edit there that could alter the profile of a disease. And second, what came shortly after that was the understanding that the regulation of expression of fetal hemoglobin was largely driven in red blood cells or in developing red blood cells in the bone marrow by a transcription factor called BCL11A, and that what controlled BCL11A expression in those developing red cells was a very specific enhancer element in the promoter. And so you put these two things together now, the development of a technology platform and the target, and you could say, well, if you could edit that site in the BCL11A erythroid-specific enhancer, you could alter the expression of BCL11A in developing cells of the bone marrow and turn on fetal globin expression that we've known since 1947 is protective in the disease. And so that was really the story behind what became Casgevy, was this long history and understanding of the biology of the disease. And then the relatively rapid development of additional molecular features and the CRISPR-Cas9 editing technology that you could use to develop a novel therapeutic for. So I think it's this really nice intersection of, biology and emerging technology that rapidly translated into the clinic, which I think for rare diseases is something that I think is common for what both Michelle and David are pursuing as well.
Mm-hmm. So you would characterize Casgevy as a gene therapy. Is that accurate?
It's a cellular therapy that uses a CRISPR-Cas9 gene editing approach.
Mm-hmm.
And the way in which this is done is we take the stem cells from a patient,
mm-hmm.
they are edited, and then returned to the patient as a cellular gene-edited product.
Got it. And, it's interesting you're talking about sort of this long history and how we've known, the underlying basis for sickle cell anemia for a long time. I'm curious if you could give your perspective just, through this journey, did you sort of learn about, I guess, gene and cell therapies overall in a way that, that general approach could be applied more broadly?
Yeah, I mean, I'll speak to, some anecdotal events in my own life. I originally got interested in science and research and medicine as an undergraduate student, and that was serendipity because I had an opportunity to work as an undergraduate in a research lab that was directed by a very inspirational virologist who was interested in using viruses for gene therapy. And I think that was the original vision of what genetic therapies could be, were viral vector based. And there have been many such developments using them and continue to be. I think the interest and the vision of that has been going on for a very long time. For me, this was in the 1980s, but these were theories and platforms that were developed in the 1970s as well. So I think there's been a long history of development of novel types of therapy, including genetic medicines, for a long time. It's really the refinement of the technology and its specific application for a disease. And that really requires an understanding of the cause of the disease to understand how to use these tools to get to a potential treatment option.
Excellent. Thank you. So switching gears a little bit. So David, your approach, so, Bill was sort of developing new therapies. The medications that you're looking at, they're already out there. They're in the world. We just don't know necessarily what they do. Can you talk a little bit about how you're trying to unlock the potential of these drugs through your technologies at Every Cure? And, have you discovered anything surprising in this process?
Sure. So there are about 4,000 FDA-approved drugs, which we as humanity should be so proud of. I mean, it's incredible. 4,000 drugs, and they're approved for about 4,000 diseases, which seriously, the scientists that are watching, amazing. But there are still 14,500 diseases that don't have a single approved therapy. And it is so amazing the work that is being done and needs to continue to be done to develop new drugs for many of those 14,000 diseases that don't have any therapies. But many of those 4,000 existing drugs can treat some or many of these diseases that don't have any treatments. And you mentioned that we're looking for drugs where we don't know what else they could be used for, and that is the case. We are looking to identify new uses for drugs that the world doesn't know about. But what's been really surprising is that we found a lot of additional uses for approved drugs that the world already knows about, but there's some systemic barrier that's preventing that drug from reaching patients. Usually it's that the drug is cheap, old, and generic, so there's no path forward to market the drug or to commercialize using it in a new disease area. And that's, I think, probably been the biggest surprise. And I'll share a couple examples. One of them is with TNF inhibitors for a rare disease called DATA2 syndrome. So DATA2 is a horrible condition where kids start having strokes soon after birth, and they usually die in their teenage years because they have hundreds of strokes. And about 20 years ago, a researcher, or not a researcher, a physician, decided to try a TNF inhibitor in one of his DATA2 patients. And it was because he had leftover TNF inhibitor in his syringe. He had just treated one patient, he decided to treat another patient. And then that patient stopped having strokes. And over the course of the next few years, he saw more DATA2 patients. He gave them TNF inhibitors, and they didn't have strokes either. It was 15 years from that initial observation before the DATA2 Foundation, led by Chip Chambers, began to put data together around this. And then Chip came to us, and we started putting together treatment guidelines, so that way everyone with DATA2 syndrome could be recommended to actually get a TNF inhibitor, to where we are now 20 years later. Now every kid with DATA2 is getting a TNF inhibitor. But because there wasn't a commercial path forward, because TNF inhibitors were being utilized for more common diseases as opposed to a rare disease like DATA2, the world knew. But 20 years passed and thousands of kids died from strokes, even though we knew a good treatment for them. So that is a really extreme example, but there are many more examples like that. One that we've come across recently and scored really well on our platform is a drug called leucovorin for a rare percentage of children who have autism, where we say that they have autism spectrum disorder, but really they have antibodies against the folate receptor, so they get no folate into their brain. We call it autism spectrum disorder, but really it's just a cerebral folate deficiency. But if you give high-dose leucovorin, which is a reduced form of folate, it can get into the brain through a folate transporter. And there have been 4 randomized controlled trials showing that leucovorin can improve speech and verbal communication, a number of other ASD-related symptoms in these kids, but no one's getting it. And it's because leucovorin is cheap and old and been around for decades. And so these are literally hiding in plain sight. And in these couple cases I mentioned, we didn't have to do any clinical trials at EveryCure. We literally just had to use our platform to uncover them. And so at EveryCure, we're using this platform to quantify the likelihood of every drug to treat every disease. Sometimes they're predictions, like you mentioned, but sometimes we're just uncovering work that someone else did but just hasn't taken it to the finish line. So we'll do lab work, we do clinical trials, and in some cases like leucovorin, we put spotlights on these opportunities and really encourage the off-label use of these medicines.
And maybe it'll be beyond me, but can you explain in a nutshell sort of how your platform works?
Sure. So we utilize what are called biomedical knowledge graphs. We have a handful of knowledge graphs that we use, which are visual representations of what the world knows about all of human biology and all of medicine. So you can imagine there's somewhere between these different graphs between 3 and 10 million nodes. So a node in a graph is a biomedical concept that is then connected by edges. So for example, with Castleman disease might be is a node, and it has an edge to interleukin 6 because Castleman disease is associated with increased interleukin 6. Interleukin 6 has an edge to siltuximab because siltuximab inhibits interleukin 6. So now you have a triplet between Castleman's, IL-6, and siltuximab. Imagine doing that across all of human biology and all of human knowledge. Every node, every edge, like I said, somewhere around 5 million nodes, depending on your graph, and tens of millions of edges connecting each of these concepts. So now we have like a 2D representation of what the world knows about human biology and all of medicine. And then we train algorithms on known treats relationships, meaning examples like siltuximab for Castleman's. We train the algorithm and saying, this is an example where the drug works for this disease. And we also train the algorithm on these are examples where this drug does not work for this disease. And then we unleash the algorithm on the rest of the knowledge graph to come up with a score from 0 to 1. We use an algorithm called XGBoost. And if the pattern of connections in this new drug disease connection, which could be folinic acid for pancreatic cancer, you name it, it's going to give us a score. If it's not a good match, if it doesn't look like a known treatment relationship, it's going to be closer to 0. And if it looks very similar to these other known treatment relationships based on the pattern of connections and the intermediary nodes, then it'll get a high score. And then we at EveryCure, our team then looks at the highest scores and says, wow, this drug for this disease looks really promising. Let's do laboratory work. Let's do clinical trials, and let's move them forward.
Wow. See, I kind of got that a little bit. Great. We're going to move on to tRNA, one of my favorite molecules, I must say. It's lovely. Michelle, can you tell me a little bit about how, like, why tRNA? And I thought it was interesting when you were introducing it that you could, one approach could have large ramifications and be able to treat lots of disease. Could you kind of explain how it works and why it could be so semi-universal?
So first of all, bonus points to you, Erica, for knowing what a tRNA is, because oftentimes people forget what a tRNA is from our Biology 101 days. But yeah, so I mean, just maybe to talk about what a tRNA is, and then we can go into why it's, important and how we think it can be used is, so the tRNA plays a critical role in the protein translation process. So I'm sure folks have heard the central dogma about how DNA codes for an mRNA, which codes then for all the amino acids that make up a protein. And if on the mRNA, each amino acid is coded for by a 3-letter code that makes up each and every amino acid, and the chain of amino acids make up the proteins, and the proteins are what keep us healthy and alive. So the tRNA plays this critical role where it actually deciphers the code for each and every amino acid on that mRNA coding sequence, and then finds that corresponding amino acid in the surrounding cellular environment and transfers the amino acid to the growing polypeptide chain, hence the name of a transfer RNA, because it plays this important transferring role of the amino acids. Now, what happens oftentimes when you have different diseases. So David, you were the first person that actually mentioned 14,000 different diseases. I've always said somewhere between 6,000 and 10,000. I think that's kind of part of the issues. I don't know if anybody actually knows how many discrete diseases there are, but it's a lot. And many of these diseases, over 80% of these diseases, have a genetic component to them. And when you think about all of the modern technology we have access to today, so not just small molecules, but genetic medicine, cellular therapies, each one, whether it's a gene therapy, a gene editing, an mRNA type approach, each one of these approaches really must take a disease by disease or gene by gene type strategy to tackle those conditions. And that's because they're very specific to a certain gene or a certain protein, and that's how those mechanisms work. Now, of course, when you have, I guess, somewhere between 6,000 and 14,000 of these different diseases, and we're trying to tackle them one at a time, it's going to be an eternity, really, before we make a dent in them. And often the way that drug development works is you start in one disease, you prove that it works, and then you start in the next one. And it's this sequential iterative process, disease by disease at a time. So we're taking a very different look at that problem of so many different diseases, one at a time, and taking a much broader approach by identifying patients that have common mutations across all of those different diseases. And we're starting in the first instance with tackling what's called a premature termination codon, or a nonsense mutation. And that's where one of these codes for an amino acid gets mutated and codes for a premature stop instead. And what happens is that the translation process ends too early. You've got shortened protein, which is dysfunctional or non-functional in nature, and that's what causes disease at the end of the day. The same exact premature termination codon mutations are seen in thousands of different diseases. And when we're looking at engineering tRNAs that have the ability to read through these premature termination codons, so instead of the protein translation stopping at that mutation, it actually knows what amino acid should have been coded for, finds that amino acid, restores the protein translation process, and it continues to the point where you have a full-length functional protein at the end of the day. And because we see the same exact mutations across many different diseases, we can use the exact same engineered tRNA across dozens, hundreds, perhaps even more different diseases, because we're taking a very mutation-specific approach, not a disease-specific approach. In fact, it's actually disease agnostic. So the tRNA does the same thing naturally, as well as our engineered tRNAs have the same function. The biology is exactly the same, regardless of the gene that's affected, regardless of the protein that's being encoded for. And it actually is also regardless of where the mutation occurs in the coding sequence, which is, again, one of the unique features about this type of technology. And what's really exciting to us about this is the way that we're thinking about developing these medicines is not just, typically what we see is one clinical trial with one disease with one medicine. We can do one clinical trial with one medicine, but multiple diseases with patients selected by those common mutations. And what that allows is not only having a clinical trial or an intervention available for perhaps the most common of these genetic diseases, but we can bring along with that all of the ultra rare, maybe nano rare type diseases, the onesie, twosie populations, maybe that make up these 14,000 different diseases that you mentioned, David, because many of those ultra rare and nano rare diseases are too small. They just don't fit the model for a disease by disease strategy, and will unfortunately likely never have a novel innovation sort of dedicated for it. This may allow the opportunity to bring along all those patients with a mutation-specific strategy independent of disease, and hopefully be able to offer some optimism and hope for those populations. And I can speak to, how important that is for families. When my son was diagnosed a number of years ago, especially as somebody who's been in the industry for a couple of decades, the first thing I did was start to explore what clinical trials were ongoing that he might be eligible for. And I was really horrified when I realized that there were 0 interventional clinical trials that he was eligible for. And my heart just sank at sort of the possibility of, gosh, is he really one of these overlooked individuals that will never have anything other than, I'd say very a standard of care that hasn't changed for several decades and is really not acceptable. So I know what it's like to be one of those families, and I hope that with this type of technology that we're developing here at Alltrna, that other families like ours wouldn't have to go through that same type of feeling.
That is so cool. So is the actual sort of therapeutic agent in these cases like the tRNA? So is it like a small molecule type drug approach, or is it a gene therapy? I wasn't entirely clear on sort of what the agent is.
So it's an oligonucleotide, so
Okay.
an RNA-based technology. I would put it into a genetic medicine category. It's not a small molecule, but it's also not a gene therapy, right? So I guess it sort of fits into this own category. It's its own new class of therapeutics.
Wow. So what a great group. Oh my gosh. You're all doing different and amazing things that are having a real impact and have bringing a lot of hope. Can we talk a little bit about, you're all taking these different approaches. Is that good that we're trying all these different approaches to the rare disease problem, right? And I wonder if you could maybe, Bill, if you want to start, is there benefits to be gained from having many different strategies for tackling rare disease?
Yeah, I think that's a great question. And yeah, I think what's really exciting, particularly now for a lot of rare diseases, is that the technology options that we have available to us are exponentially increasing. 10 years ago, we didn't have CRISPR-Cas9 gene editing. The technology that Michelle is referring to is newly being used as well. I can speak a little bit for, at Vertex, what we do is I think anecdotally important as an example for this, which is we look for diseases that are severe, that are life-limiting, life-threatening, that we have an understanding of the causal biology of, meaning that there is a validated target that we think relates to clinical outcomes. The example is in sickle cell disease, where we understood that the BCL11A erythroid-specific enhancer target could increase fetal hemoglobin, which we knew from well over 50 years of natural history clinical research translated to protection from disease complications. If you then take the next step of what do you need to do to address that target, it could be any number of different tools. For us, it ended up being CRISPR-Cas9 gene editing for sickle cell disease, but for another disease, that may not be the right technology approach. And it could be something very different, tRNA. It could be a small molecule. It could be a biologic. It could be any number of other therapeutic modalities. At Vertex, our approach is really to take whatever the right tool is to hit the biology to affect an outcome. And I think writ large, I think all diseases, and particularly in the rare disease space, can be a little bit different and may need a different approach to hit the underlying biology. And that's, I think, probably the most platform agnostic way to think about it. There will be common types of approaches where one platform makes a lot of sense for a lot of different diseases. But for some, that may not be the right approach. And I think what's really exciting right now in the space is that there's so many different technologies that are now emerging and available that we can use to help people living with rare diseases.
Great. David, Michelle,
I guess maybe the only other, yeah, I would just say, I mean, the number 14,000 different diseases, I mean, you can imagine the diversity amongst them is massive. So there's never going to be like one single way in which all of those different diseases can be tackled. So yes, I do think that the responsibility is on us as scientists and, curious individuals that really want to push the understanding forward to really explore many different options so that patients have as many shots on goal as possible.
I completely agree, Michelle. I hate to say this, it's actually 18,000 diseases. It's just 14,000 that don't have treatments. So we got 4,000 out of 18,000 figured out. So it's actually bigger than that 14,000 number. But there's 14,000 with 0 treatments. But I totally agree with both you and Bill. And I think that we need to be taking as many shots on goal as possible. And on top of that, we need to recognize that for drugs that have already been developed and they are on the market, as Michelle said, there's this sequential, okay, we found a disease for it, let's find another indication. But we know that as soon as that drug begins to get close to patent exclusivity or losing patent exclusivity, then all research stops. And so even when we make new discoveries that GVHD involves interleukin-6 signaling, if that interleukin-6 inhibitor like tocilizumab is towards the end of its life cycle, the work's not going to be done to then look at toci for GVHD, for example. I'm just throwing out a random example. And so there's a lot of these opportunities where there's really good mechanism, but the incentives aren't there. So you have this world where someone is suffering, and then there's this drug that's actually at their neighborhood pharmacy that could help them, but the world isn't making the connections. And I love that Michelle made the point around her disease agnostic platform. And that's what we need in addition to the very disease specific. So we need very disease specific work, but then we also need these disease agnostic approaches. And we describe what Evercure is doing as being disease and drug agnostic, where, we don't have any drugs in our pipelines. We don't care what the drug is. And we don't have any diseases that we're focused on. We don't care what the disease is. We just want to help people. And then if you can quantify the best connections between every drug and every disease, and you don't care, again, whether it's one disease or one particular group of people, and you just want to help people, then all of a sudden these hidden cures just begin to emerge, and it's so fun.
Great, thank you. And we've been focusing a lot on rare diseases, and I'm wondering, I guess some of you are looking at disease agnostic approaches, so not necessarily only rare diseases, but is there anything different about research in rare diseases that has led you sort of down your particular research journeys or challenges that are particular to rare disease communities that you're trying to address through your approaches? Let's go with Michelle first.
Yeah, I mean, so I would say, I think that certainly our company, Alltrna, focusing on the tRNA space, we really stand on the shoulders of giants. In the last couple of years, ourselves having learned so much from the advancements that have been made in RNA biology and RNA therapeutics across the board, with the emergence of siRNAs, mRNAs, like we really have a much, much, much better understanding about how to leverage this biology and really capitalize on the biology and turn it into a therapeutic, which has been, a great inspiration for us. Rare diseases are very different than common diseases. And I would say you pick up on some of the challenges, and yeah, these are not easy spaces to go into. You think about like the diversity of these different diseases. Many of these diseases that, that we've been talking about are really not well characterized or well understood. So Bill mentioned about like a deep understanding of the biology of sickle cell and beta thalassemia, leading to some of the innovations in that space, and all of the sort of AI work, David, that you're doing to really understand the kind of key issues within those discrete diseases. I mean, it really takes a fundamental understanding of that to be able to think then about, here's the problem, here are the solutions. And many of these diseases, I'd say even most of these diseases, aren't well characterized. And even within those diseases, there's a tremendous amount of diversity between the phenotypes that patients have that makes it very difficult to really even, sort of have a universal understanding on how to tackle that. I think that's one of the key challenges that has sort of gotten in the way of advancements within this space, not to mention the smaller patient populations, which just in general have made things a little bit more difficult from a drug development perspective, and then the attention from a drug discovery perspective. But that's why I think, some of these technologies that we're talking about today are so critical, because they really help alleviate or try to address some of those challenges and still not have it become a limitation in advancements moving forward.
Anything to add, Bill or David, on that?
I would say I think, and I think both Michelle and David have articulated this extraordinarily well, is that the really compelling thing in rare diseases is that these are often ones where patients have very few, if any, treatment options. The burden and the unmet need is enormous, and that is extremely compelling, because we feel, I think, as a society, that we should have a responsibility to do better. And we hear it from patients and families all the time, and that drives, I think, a lot of this forward. But I think the key thing is that there are the three key elements that in rare disease are, you could call them challenges, or you could say are the most opportunities. There's the basic science research to understand the disease. There's the education and awareness and the clinical expertise that's needed to manage the disease. Often these are rare diseases where the access to a care pathway with an experienced clinician isn't really available in many places. And the third is the will to develop new treatment options. And I think David has articulated some of that very well, that there are a lot of challenges, which I think he's converted into opportunities, which I think is phenomenal. So for rare diseases, I think those three aspects of research, clinical care, and new therapies is what makes it really compelling, because at the end of the day, it's really about helping people who don't have any other treatment options.
Great, thank you. David, we're going to move to you. So as both a scientist and a patient, can you talk a little bit about how your personal journey has influenced your approach to developing treatments for rare disease?
Sure, it's changed everything. I think that, if I had to focus in on a few, I think that one is this tremendous sense of urgency that of course I had when I was trying to find a drug to save my life. Fortunately, I've been in remission for a long time. So the sense of urgency is not to find a drug for me. The sense of urgency is to find drugs for other people who are currently in the same exact situation that I used to be in, which is I was suffering, dying, while there was a cure that could have helped me, and we just didn't know it yet. So I have this incredible sense of urgency that hasn't dropped at all since I found a drug for myself. It's almost even grown, because we've literally helped thousands of people across about 14 diseases where we found these repurposed drugs for. And so it's just grown and grown and grown the more and more people we've helped, because it's made us realize, oh my gosh, if we hadn't done this, like, these people would not have been able to be helped. And so I think sense of urgency is one. I think that being very focused on on impact with everything we do. I love and I appreciate and I value basic science, and I also always want to be thinking about how can that basic science be translated. Let's not let it get published in a journal and then no one does anything about it. And I'd love to give an anecdote about that after I maybe share a third one. And I think the third one, that being a patient, I think it's helped me to think a lot about denominators. You know, we were talking, Michelle, 18,000, 14,000. I think that we can oftentimes get very focused on our disease or our task at hand, our company, our thing. But we oftentimes forget the denominator about all those other things that aren't being tackled. And of course, I know what it's like to be any one of those things, those diseases that aren't being tackled. And so I think that it's about, you know, sense of urgency. It's about making sure we're always focused on the end, which is drug in mouth, drug in vein, helping patient. And then also the denominator, all those people where maybe someone isn't thinking about them and maybe we could. And I'll just share a really quick example for the second one. And that's this idea about always translating. And that is that we had a patient come to our center with a horrible cancer called metastatic angiosarcoma, and he had failed to respond to the 2 chemotherapies that are recommended. And his doctor told him, "This is it. We're going to transfer you to hospice care. You know, you probably have 3 months or so before your body's riddled with these tumors and you're going to die." And he reached out to our center and he said, "You know, David, I heard that you guys found a drug for your disease. Like, you know, you repurposed something. Like, could there be something else for my angiosarcoma?" And we did the simplest thing, Erica. We literally went to PubMed, the database of papers, and we searched angiosarcoma treatment. And we came across this paper that had been published 3 years earlier in PLOS One, so not a particularly you know, broadly read journal, but published. And in that paper, there were 5 patients with angiosarcoma where PD-L1 expression was measured, and 4 out of the 5 patients had increased PD-L1 expression. Now we're 3 years later, 2016, and PD-L1 expression, of course, is a tremendous biomarker for whether you respond to a PD-1 inhibitor or not. But 3 years have now passed by between this paper being published and no one in the world being treated with a PD-1 inhibitor for angiosarcoma. And so we said, well, why don't we just test this patient, Michael, to see if he has increased PD-L1 expression? And well, first off, his doctor was completely against it because this drug had never been used before for his cancer. And he said, "I'm not going to test for it because I wouldn't prescribe it anyway." Anyway, we tested for it and it came back blazingly positive. And we got Michael on a PD-1 inhibitor for the first time ever for anyone with angiosarcoma. And this past April marked 8 years that he's been in remission. Last month, he walked his son down the aisle on his wedding day. And everyone was dying within a year. And now about a third of people live full lives, or at least thus far, it looks like extended lives on pembrolizumab for their angiosarcoma. I bring this up as an example of always thinking translationally. Those researchers who studied those 5 tumors and found 4 out of 5 of them had increased PD-L1 expression, they did their job, they did their science, they published it in a journal, and then they went and they started doing some other basic science work. And no one was picking up this insight to then move it into a patient. We have to make sure that there's someone picking up the scraps and making sure that like these insights aren't falling through the cracks. And that's the value of something like a biomedical knowledge graph, because every one of those insights, every single node, every single edge is inside this graph, and it gives us the opportunity to find those hidden gems that are literally just hiding there in plain sight.
Well, I know where I'm going when I get sick. It's like, hello, remember me? Thank you for sharing that. Now I've like lost track because I'm like a little misty. Michelle, also a member of the rare disease community through your child. How has that been a part of, I mean, a big part of your motivation, and but has it shaped your particular approach in any way through Alltrna, and is that condition part of your goal for treating?
Yeah, absolutely. I mean, I would say, you know, throughout my 20+ year career, I've always been a very patient-focused leader, but I don't think I really understood that as well as I did when my family was personally affected, and we were those patients that, you know, certainly was hopeful for novel innovation. The thing that I think is so important to really recognize, especially in the rare disease community, is how critical that patient voice is in not just sort of understanding the disease, but also driving the science and the momentum forward for those particular diseases. I would say when my son was first diagnosed, the first thing I did was actually reach out to the Duchenne advocacy groups. And, that's where I got my training to really understand the disease, understand what was important, understand what was being explored in that space. And I was really taken aback by the responsibility that they had and they take seriously to not only educate the community of affected families like my own, but also educate scientists about what's important and educate and really inspire and oftentimes fuel sort of dollars into research specific for that disease. Truly amazing. But that's not the only one. We see this over and over with so many different diseases, whether it's Rett syndrome or in the organic acidemias. These groups are doing the exact same thing. And honestly, it comes from a place of necessity. When you have a child who, as was essentially our experience, but most of these families are essentially told there's no medicine for them. Go home, love your child for as much time as you possibly have with them. Many of these families refuse to take no for an answer. And I applaud them and I am inspired by them. And every single day, I think about those families and those groups who have made such a dramatic difference in pushing science and pushing research forward in an incredible way that just would not have happened otherwise. They're really the heroes at the end of the day in all of this, not to mention then offering up either themselves or their children to really be the tests to see if medicines are going to work for the future generations that have that disease. So I would say for me, I take this incredibly seriously at Alltrna. One of our company core values is patients deserve better. And that really comes from hearing this narrative over and over again of just going home and loving my kid for as much time as possible isn't good enough. We need to be doing more. And so really being inspired every day to do more, to do better on behalf of these families and these individuals who really just deserve it. And so, yeah, I will say even in my role as CEO, we get contacted by patients and patient groups all the time. And, every single week, I take every single call. I answer every single email myself personally. That's how important it is to really just listen to their stories, be humbled by them, be inspired by them, and to really fuel that, the work that we do at Alltrna into it. And then part of my job then is really communicating that to the rest of the organization so that they hear it and they sense it too. And so that every single, person matters, every single day matters, and that we really have to make the most of what we're doing in order for us to benefit these individuals.
Oh, thank you for sharing that. And Bill, you were talking earlier about sort of what drew you to sickle cell disease and in this community, that you had some interactions early in your career. Could you talk a little bit more about that and how it helped shape your work, and sort of what is the success of, I'm going to do it again, Casgevy. I'm trying.
Casgevy.
Casgevy.
It's a soft G.
Soft G, Casgevy. So what does this success with Casgevy mean for you?
Yeah, I mean, I think the way that David and Michelle have articulated it from the patient perspective is really what drives a lot of this. And I think, Michelle, even earlier, you made a statement about, that you often felt like you were standing on the shoulders of giants. And what we often think about that is our research and clinical forebears. But really, that means patients, because for many of these diseases, it's patients and families who have been advocating for a very long time, in fact, demanding that we should have better treatment options. And I think that's exactly it. From my own personal experience, when I was in the clinic, for sickle cell disease, there were very few treatment options, and there still are very few treatment options, more maybe now than there were then. And the way the disease was managed was essentially the same way that it was managed in 1910 when the disease was first described, which is patients have excruciating pain because of blockages in their blood vessels, which I always make the analogy that it's like having a mini heart attack just all over the body. And the way that we would manage it is we'd use a little pain medication, some IV hydration, and warmth. And that's essentially the way that those events are managed today. And so when you're in the clinic in this era and what you have to say to patients, the only thing I have for you is to ask if you drank enough water today is really, and I think, Michelle, what you were speaking to, that's not what people are looking for, and that's not what they will tolerate. And they, and I think we all respond to that. Patients have been demanding equitable access to care and equitable development of new therapeutics for a very long time. And that was certainly true from my experience. I think, when we get to the other end of it and we have a treatment option, we tend to develop drugs in terms of a primary endpoint. Thalassemia, you no longer have some transfusions, and in sickle cell disease, you no longer have these painful vaso-occlusive events, perhaps. But what does that really mean for patients and families? And I think when you can see that translatability about what it means for their lives, and then, the story, David, that you told about the individual being able to, years later, walk his son down the aisle. I mean, this is what these outcomes mean. They're beyond a statistical number. They're really ideally changing the way that people have the opportunity to live their lives the way they would otherwise choose to. And in rare disease, I don't think the patients are asking for anything other than what is done for, I don't know how many diseases there are that actually have a treatment option relative to the 14 or 18,000 that don't. But they're really just asking, well, we should be able to develop for our rare diseases as well. And the impact of those are no less relevant. And so I think that's what really drives a lot of it. It certainly has been for me and for us at Vertex, is seeing what the outcome can be if you do have a new treatment option.
Great, thank you. I want to talk a little bit, spend a few minutes talking about access and equity with these treatments. What do you believe is the biggest challenge with ensuring equitable access to, cures to rare diseases, these really promising gene therapies and cell therapies and repurposed drugs that have been forgotten by the wayside? So maybe, David, we can start with you. What do you believe are the biggest challenges, and how might they be overcome to get more people access to these life-changing treatments?
Yeah, so in our camp, which is around obviously repurposed drugs, the barrier is not cost. The barrier is awareness and understanding about the additional use. So we recently came across data around lidocaine as a treatment pre-surgical excision in women with breast cancer. So if you inject lidocaine directly in the tumor, large randomized trial found that there was improved 5-year overall survival from a direct lidocaine injection due to a really, really interesting mechanism. But no one's injecting lidocaine into tumors right now because lidocaine is about the cheapest. There's no profitability from like an extra squirt of lidocaine, so there's no advantage for anyone to market that. And we just have to recognize that within our system, that means that no one's going to do it unless someone starts raising awareness and advocating for lidocaine. I actually got an email yesterday from a surgical oncologist who said he just injected lidocaine in his first patient yesterday. But so I think for what we do, it's all about awareness and education, because they're cheap old drugs. I promise you, insurance companies do not turn down drugs that cost $1 a day. They just don't. And so it has nothing to do with insurance or cost of drug. For the vast majority of drugs, over 80% of FDA approved drugs are already generic. And by definition, there's multiple manufacturers, the cost is low. There's no problem getting those covered. The problem is interest and education and awareness. And if you can handle the interest and education, at least in the United States, where the majority of our citizens are insured, then you're going to get these things paid for.
Mm-hmm. Bill, what are your thoughts? You have sort of probably the opposite issue. Some of these emerging treatments are some of the most expensive, medications in the world, and there's a large amount of need for them. Any thoughts on access?
Yeah, I mean, I have a few. I mean, obviously, I'm on the clinical development side, so the access things are a little bit outside. But my observation about it is that, and particularly, and I think this is true, as David talks about it too, there are a lot of different stakeholders that are necessary to get access for treatment options. Those are, there's governmental ones, there's insurance, there's patient advocates, physicians, and the companies as well. I mean, I think the key thing is that we need to have frameworks where all of those stakeholders can work together. And particularly for some of the new emerging technologies that require different paradigms, I think that is more important than ever. Because patients we know are waiting 14,000 diseases. That boggles my mind, that number, even 18,000. I mean, that's amazing. We do need to have our stakeholders be able to get together to be able to ensure that patients have access to the right treatments that can potentially help them. And whatever those modalities are, whether we're repurposing something, developing a new technology for a disease that has very few people, or whatever it is, that's a common theme for all of them, I think. And that's not easy to do. Moving government is, a long-term type of endeavor. I think, the will that we have to have as a society, though, to say that that's what we expect and that's the framework that we need is, to me, the starting place. And I think that often comes from patient advocates who are the initiators of doing that.
Mm-hmm. And Michelle, you have sort of more forward-looking. Is there anything about sort of how you're developing this whole platform technology that you're sort of thinking, at the end, how will patients access this? Or is there anything sort of long-term that you're sort of building now, thinking in the future about access?
Yeah, I mean, I think that we all need to really recognize that that drug development is hard. Drug discovery is complex, and manufacturing as well, right? And none of these things are cheap. But what's also, I think, really important, you talk about having these different frameworks and paradigms, and I actually think that's really one of the key considerations for us at Alltrna, is that we have to break free from this paradigm of a single drug for a single disease, because that's the most expensive way of innovation. So this, in our platform, as we've discussed already, the idea of having single drugs across many diseases really helps tackle a lot of that. So instead of having to do drug discovery for thousands of diseases, it's drug discovery for a few. The same thing also, instead of having to pay for clinical trials for each and every disease over and over for different medicines, one clinical trial perhaps across many different diseases. These all have a major impact in terms of the overall efficiencies that we can build into the system with platforms like these. And so hopefully that will create a future world where access to innovation will be a bit more equitable, but definitely there's a lot of work that we need to do through the entire system to address this.
Yeah. So for our audience, is there, we've talked a lot about sort of the patient role, the family role in sort of the overall development of treatments for rare disease and how impactful that can be. Just based on your experience, I was hoping we could go around for our viewers. Is there any sort of call to action that you would suggest? Like, what could people, the rare disease communities, what are effective actions they could take to help drive curative therapies forward? Or what have you observed in your experience? David, you want to go?
Sure. So I shared earlier about the AI platform that we've built that's quantifying every drug versus every disease. We also have a very simple, I call it maybe a bypass pathway, and that's where patients can go to everycure.org/insights, and they can share about a drug that they've received from their doctor and whether it's helped or not helped. And we always love when these insights come from our AI platform, but we also love when they come from patients who can share that this drug worked for them. Over 20% of prescriptions written every day in the US are off-label. So doctors are prescribing things for diseases that they're not approved for all day, every day, all over the United States. Like I said, it's between 20 and 30% of all prescriptions. So when patients do receive something off-label, or when a doctor does prescribe something off-label, and there's something interesting that happens, something maybe they've noticed some improvement in their symptoms, maybe there's some early insight. We want to hear from you. We want you to come to everycure.org/insights and let us know. We're going to look at our platform score, see what the score is from 0 to 1 from our AI platform, but we're also going to note that it helped a real patient, and that's going to help to flag it so we make sure that we're moving it up in our list.
All right. Bill?
Maybe I'll just jump in here. Right?
Yeah, sure.
So I I think that there's like one big call to action is, I will say when somebody gets a diagnosis like this and you feel like you're the only one in the world with this disease or you've never heard of it before, it can feel very isolating and extremely intimidating. But the reality is that you are not alone. Rare diseases are actually very, very common. 1 in 10 people have a rare disease. It's very common. And one person can make a big difference. So find your people, find your network. If the network doesn't exist, build your network, just like David did. And, the collaborations that can be brought together to really tackle these difficult problems can move mountains. So you don't have to settle for just wait for something to happen. There are things that you can do. You are powerful and you can make a lot of things happen. And I think that's, certainly something that I've seen from many of the families that I've interacted with. And I think that's something that I think we all can have, have learnings from.
Thank you. Anything to add, Bill? Yeah.
I agree. I agree with that. That was eloquently said. I mean, I think the message is keep doing what you're doing and don't give up hope, and it matters, right? It may not be today and it may not be tomorrow, but we still have hope for that. I would say, we had a public advisory committee meeting with the FDA for Casgevy. And within that, there is a time for public comment. And there was a patient advocate there who, and this is recorded, I think probably still on the FDA website, who made a comment that, sickle cell disease had been in her family for generations. And what her hope is that she may not be the last that would have a child with sickle cell disease, but hopefully the last who would suffer as much. And I think the compelling thing is to keep demanding, keep advocating, and keep going, because the hope for future treatment options, we hope we can respond to over time, and we really need those voices to help.
Well, unfortunately, we'll have to stop there as we've run out of time. Many thanks to our panelists for being with us today and just for the incredible work you are all doing. I learned so much, and I hope our audience did as well. A reminder to our viewers that you can see a recording of this webinar as well as all of the previous events in the series at science.org/webinars. If you'd like to send your thoughts, please email us at webinar@AAAS.org. Also, viewers, I wanted to let you know in just a moment, you'll be invited to fill out a brief survey. Your responses are so important and will help guide our future conversations on rare disease. Thank you once again to our fantastic panel and to Foundation Ipsen for enabling this conversation through their kind sponsorship. Goodbye, everyone.